Hi, everyone, and welcome to the 52nd seminar presented by the Centre of Research Excellence in Aphasia, Recovery and Rehabilitation. I'm Dr. John Pearce, postdoctoral researcher at the Aphasia CRE and your host for today. Before we do anything, I'd like to acknowledge the traditional owners of the lands where we live and work in Australia. I'm speaking to you from Boonwurrung land, and the Aphasia CRE office is situated on Wurundjeri land. And we honour their elders past, present and emerging, and extend our respect to all Aboriginal and Torres Strait Islander people joining us today. Very privileged today to have Dr. Caterina Breitenstein with us this evening, or this morning for Caterina, to discuss her expertise in designing trials for aphasia. And I'm going to share more about Caterina's background shortly, but I'll just first outline how to submit your questions. So do feel free to submit questions for Caterina using that Q&A button down the bottom of your screen, but avoid using the chat. You're welcome to submit questions at any point during the presentation, and that means other people can like the questions they're most interested in. So I'll prioritise the most popular ones when we begin the Q&A. We just ask that you keep your questions brief and focused and avoid submitting comments. We've reserved some time at the end of Caterina's talk to address as many questions as we can in that time. So it's my pleasure now to introduce today's guest speaker, Dr. Caterina Breitenstein. Caterina is a trained clinical psychologist and now a senior researcher in aphasia rehabilitation, specialising in the measurement of treatment outcome and in randomised controlled trials to determine the efficacy of behavioural, pharmacological and brain stimulation treatments. She was the PI of the FCE T2EC healthcare trial on the effectiveness of intensive speech therapy for chronic aphasia. For some time now, Caterina's research activities have also included qualitative aspects, particularly on the research priorities of people with aphasia regarding aphasia therapy and its outcome assessment. Before Caterina chose aphasia as the focus of her research over 20 years ago, she investigated communication behaviour in chronic pain patients and their partners, emotional prosody in Parkinson's patients, and developed language learning models based on associative learning principles. Caterina has a strong affinity for statistics and is passionate about improving the methodological quality of aphasia research studies. She was Deputy Chair of the Collaboration of Aphasia Trialists and Secretary of German Aphasia Research Society until 2024. Thanks for joining us quite early for you, Caterina.
Yeah, thank you very much, John. So I'm going to share my slides. Okay, so I'm hopeful
you might just, oh, there we go. No, looking good.
Okay. There may be a time lag here from Germany to Australia. Yes, so thank you very much for the opportunity to present here today. So I'll be talking about designing trials for aphasia, the do's and don'ts. And I embedded, references and resources into my presentation with a QR code. So you can just scan them and then, the reference or resource should pop up. I first want to express my huge thanks to my statistics heroes, so my mentors in all statistical questions, and these are Professor Peter Matus, who is a biostatistician, and Klaus Wilmes here, both from Germany, and Klaus Wilmes is probably best known to all of you because he developed the Archon aphasia test. So huge thanks to these two, and a lot of what I say today is probably I learned from them. I first want to explain why I'm such a big advocate of high methodological quality. I'm doing that actually for the benefits of people with aphasia, because I think it's unethical to share information with people with aphasia that is not based on solid methodological grounds. And in my role as Deputy Chair of CATS, the Collaboration of Aphasia Trialists, together with Marianne Brady, we had the mission to bring aphasia rehabilitation research up to the same methodological level as in other areas of medicine. And, we already achieved a lot over the past 10 years, and we compiled all these achievements into a forum paper, which was published earlier this year in Stroke, and you may want to take a look and see what has already been achieved. And in today's presentation, I have several learning objectives. I want you to learn the key principles and peculiarities of randomized controlled trial design. In aphasia rehabilitation research, I want you to be able to select appropriate control conditions for a given research question, to be able to select standardized but also meaningful outcome measures for your trials, and be aware of the do's and don'ts of trial design in the aphasia population. My presentation today is on clinical trial design, and I would like to begin with a scheme of different trial phases. The phasing scheme has been developed for drug development research, but is now applied, in adapted versions, basically in all fields of medicine. And the first two phases, phases 1 and 2, are considered exploratory clinical trial phases, which means you start with single case series to find out whether a new intervention, works at all, at least, in single cases. And then you move on to phase 2, where you want to find out whether your new intervention is feasible and also acceptable in a small sample, and you also collect early efficacy data. Phases 3 and 4 are the phases where you really want to confirm whether your treatment works. So, these numbers are, of course, unrealistic in aphasia rehabilitation research, but phase 3 is called an efficacy trial, where you want to find out whether your new intervention is superior or as good as another treatment or as a control condition. And phase 4 is actually the phase where you want to find out whether your new intervention also works under routine care conditions. So you basically probe the new intervention in real life. I'm only focusing on confirmatory clinical trials today, which fulfill the highest level here in the hierarchy of evidence-based medicine. So it's level 2 in the evidence hierarchy here. And only systematic reviews of randomized trials, so of such level 2 studies, have a superior evidence base. So randomized controlled trials are the backbone of treatment outcome research in the medical field. And I'll call them RCTs from now on. So I hope you're able to follow with this acronym. So RCTs are prospective studies that compare the efficacy and also the safety of a new treatment to a control condition. And what is special is that you randomize your participants to the treatment or to the control condition, as shown here. And this randomization procedure ensures that potential confounders are balanced across groups. So you basically expect having people of the same age, the same severity, and other baseline variables, in the groups that you have in the trial. And if properly designed, and if you find a positive outcome, then you can have, high confidence that the observed group differences in the outcomes are a causal effect of the treatment that you administered, and also that the effects will be replicable in a future trial, which is an important aspect of clinical trials research. And the outcomes of a sufficiently powered randomized controlled trial is also the basis for translating research findings into clinical practice. So without, sound and sufficiently powered RCT, it will not change clinical practice. So I first want to discuss the key principles of RCT, so prospective enrolling, of participants, randomized allocation to treatment and control group, blinding, and also different types of RCTs. So prospective enrolling of participants means that you have concurrent data collection in all groups and arms. So basically, the data are collected at the same time. And you do this because there are some issues with using, for example, historical control group instead of prospective enrolling of all groups at the same time. First of all, with a historical control group, you do not randomize participants to the groups, and then you have a pretty high risk that your intervention and your historical control groups differ in baseline characteristics. So one group may be older, or you have more females in one group, and so on. And there's also the time-related bias. So there may be changes, in the medical standards. The population may have changed, your diagnostic criteria may have progressed, or other measures are used. So there are maybe, critical differences between, your intervention and your historical control group. And of course, now with the corona pandemic, we also know that, life-changing events have a profound impact on our feeling. And so these life-changing events can also change responses to treatment. And that's also another reason, to have concurrent data collection. And another issue is that you may have data quality issues with your historical control group data. So you may have more missing values, or as I already mentioned, changes in measurement techniques. And a fourth reason, or advantage of prospective enrolling is that with historical control groups, your control group may be smaller than with a contemporary comparison group. And then you have a high risk of over or under-meeting treatment effects, because the risk of small studies is that, the precision in estimating your treatment effects decreases, and you may over or underestimate. So in other words, with a historical control group, you cannot be sure that the differences that you observe between your intervention group and the historical control group are solely related to the intervention. And you also risk over and underestimating of the treatment effect, so the confidence that we can have in the trial results are also decreased. Another reason why I would advise you not to use a historical control condition when planning a new trial is that you automatically drop 2 levels here in the evidence hierarchy. So from a control group in a prospectively designed randomized control trial is a level 2 trial, but as soon as you have a historical control group, you drop to level 4. So that would be really painful later on when you want to publish your results, that you may be criticized for this. So another key principle, as I said, is randomizing participants to groups. And you do this because you don't want the groups to differ systematically in relevant baseline variables, and you want the group difference to be only related to the treatment effects. And the preferred method for the random sequence generation nowadays is to let a computer tool randomly assign participants to the treatment and control group, simply, to avoid any bias. So you don't want any humans to be involved, and the person who is organizing the process is usually an external biostatistician who has no other role basically in the trial other than doing the randomization and the data analysis. So this person has no contact with therapists and participants. And for randomization, you generally use variable blockwise randomization, which means, that you have certain blocks of participants. For example, here in this sample, you have 6 in block 1 and 8 in block 2. And within these blocks, have an allocation ratio of 1:1 for the intervention and the control group. So this makes sure that you have an equal number of participants in each group. And because of the different blocks, which have variable block sizes, it is very hard to guess, what the next assignment will be. And if required, you can also stratify your randomization by covariates. For example, you can have, very severe cases, in one strata and mild to moderate cases in the other strata. And then you can separately randomize them. And that makes sure that you have, also comparable sample sizes in the subgroups of your strata. Allocation concealment is another issue or another key principle of randomized controlled trials. And the latest development is that you let a randomization tool compute the group allocation after the participant was enrolled, so that there's no list available where, anyone could be tempted to look at. So, you include the patient or the participant in the study, and then the biostatistician hits the computer button, and then the group allocation is computed. So, to really decrease the bias to a minimum. So very important to not involve investigators or trial staff in the randomization and group allocation procedures, because knowledge of the next assignment could influence whether you include the next patient or exclude the next patient. So that's obviously happened in the past. And there are also reports showing that RCTs with inadequate or unclear concealment of the allocation sequence, have about 30% larger treatment effect. So this shows there's certainly a bias there if you know, or if you have a high likelihood of knowing the next assignment. So the next key principle is blinding of participants, therapists, assessors, study personnel, and also biostatistician. And blinding of participants and therapists, I think or in my view, is not feasible in aphasia trials, because, you have language interventions. And if you compare the language intervention to a non-language control intervention, everybody will notice. So it's very hard to conceal that participants are in the control group. So, but if you can still try, and not tell your patients and your therapist, what the group allocation is. But you then should at least ask the patient, the therapist, about the perceived group allocation and report that in your publication. And even if you do try to blind patients and therapists, you should always blind endpoint assessment. So your primary outcome should be evaluated by assessors who are not otherwise involved in the trial. So who are not, therapists, who are not members of the trial staff, and they should independently evaluate, your primary outcome offline. And of course, you can also blind the biostatistician during analysis by just using numbers instead of, the group names in your data file. So another key principle is that you have to select an adequate RCT type. And I don't have time today to go into all the different types of RCTs. There are parallel group designs, crossover designs, N-of-1 designs, factorial designs, cluster designs, and adaptive designs. And I'm going to focus on parallel group design today, because in my view, this is still the most frequently used RCT design. And all the other designs, have some challenges in the aphasia population. So parallel group randomized controlled trials have time parallel comparisons of 2 or more groups. And very important, not only for parallel group designs, but for all RCTs, that you formulate a specific question. So this is also endorsed in the Cochrane framework, and it's called, a PICO question. And, the letters stand for the P for population, the I for intervention, the C for comparison, and the O for outcome. And so it's crucial that you formulate a specific hypothesis for your randomized controlled trials. And here's an example. For example, for population, you have to define, which participants you are going to include, for example, in adults with chronic aphasia across all ages and severity levels. And then the intervention would be 4 weeks of intensive speech and language therapy. The comparison condition would be 4 weeks of a waiting period, which involves also usual care. And then the outcome would be that you want to show superiority in improving everyday communication after the intervention. So, and basically your trial has a positive outcome if you're able to answer this PICO question with a yes after you analyzed your data. If the answer is no, then basically means that the outcome of your trial is also negative. So for the PICO question I just gave you, the study design would look like this, that you have an aphasia intervention, usual care group, and you basically compare data from before therapy or before basically usual care period starts, and then you compare the data with a time point 4 weeks after 4 weeks of therapy or 4 weeks of usual care. And if you have a waiting list control condition, this means that the control group, after your assessment primary endpoint, also crosses over to receive the study intervention. So why do you need control conditions in the randomized control trial? The answer is that you want to control for any score changes in your outcome measures which are unrelated to the study intervention. And there are many factors contributing to score changes. I probably don't list them all here today, but I want to list a few at least. So the first factor I want to discuss is a statistical phenomenon. It's called regression to the mean. And it means that extreme outlier scores at an initial assessment move closer to the group mean, as shown here in the graph, at subsequent assessment, simply because there's always, some chance associated with such extreme outlier scores. So, without doing anything, you will have regression to the mean. And then a second factor is natural progression of the disease. So particularly early on after stroke, you have rapid spontaneous progress. And of course, your scores will also change in your outcome measures simply by spontaneous recovery. Another factor is treatment expectation by the patient. So whether the patient is optimistic that they will get better, or whether they are pessimistic because they have taken part in another trial before and didn't notice any effect. So you certainly have treatment expectations which will affect score changes. You will also have treatment expectations by therapists and research staff. So these expectations may be ventilated by verbal suggestions, so that you will tell the patients, I'm sure you will get better, for example, or, consciously or subconsciously, you will behave very friendly and interested to motivate the patient. And so in psychology, we know that, these treatment expectations by the therapists and research staff can be really strong. And so score changes may at least partly be related to these expectations. And then You also have practice effects due to the repeated language assessments, particularly if you have, a long battery of tests that you use at your assessments. In my own trials, there are usually 4 to 5 hours of assessments, for each of the time points. And here's an example from my own randomized controlled trial that John mentioned in the introduction, the FC82C trial. And here in gray, you have the data for the waiting list control group. And here are the data from before waiting to after waiting. And what you see here is a significant improvement in one of the secondary outcome measures, which is a measure of general language performance, SUPS. And simply by presenting this test twice, you see a significant increase. Fortunately for us, in the intervention group, we saw a much stronger improvement from before treatment to after, the intensive aphasia intervention that we administered, and also in the waiting list control group, once they started study intervention, you have a much steeper increase. But still, practice effects are there, and you have to control for them if you want to be able to estimate the effect of your treatment. Another factor contributing to score changes are the effects of other concomitant treatments, particularly cognitive training, that patients may receive in a rehabilitation center, may also positively impact, on the outcome of your in your assessment battery. Maybe also physiotherapy because you're more active. Music therapy can positively impact, emotional well-being. So this also has to be considered in your trial. And then another very strong factor is the social attention that you receive from therapists and research staff during a trial, and also the social interactions that these stakeholders have with the participants of the study. So you have in-person contacts, you have contacts by phone, by email, by video meeting, and of course you communicate, with the participants. So there you also have, wider, meaningful dose of language and communication stimulation just from having social contacts during the course of the trial. And so the choice of your control condition is dependent on your research questions, and you really need to know or identify your intervention's active ingredients. So what are the core ingredients that you think are working in your intervention? And then you select a control condition that lacks, these active ingredients, but is otherwise very similar to your intervention condition. And you can find a lot more details in a statement that was recently published on, selecting control interventions called the COP statements. And you can have the barcode, the QR code here in case you're interested. You can take a look at this publication. So treatment effect sizes in RCTs may depend upon the choice of the control conditions. And that has been shown elegantly in a meta-analysis on effects of control conditions in depression, in psychotherapy for depression. And what the authors observed here was that in waitlist control designs and in usual care control conditions, you have the greatest effect sizes between your psychotherapy intervention and the control condition. And for aphasia rehabilitation research, we don't yet have such results, but Swati Kiran and Marianne Brady started to work on a systematic review with meta-analysis, and I was fortunate enough to be invited to join them. And the idea is to compare effect sizes of different control conditions in aphasia RCTs. And we don't have the results yet, but I'm very keen, to find out, and I hope, that within the next months, the results will be available for this too. So, as I mentioned already, the choice of the control condition depends also on the research question, and this research question can be either that you want to find out if your treatment generally works. So, does it have efficacy or effectiveness compared to no treatment or another control condition? A second research question is that you already have a gold standard of care, but you want to find out whether a new treatment works even better or works at least as good because it's, has fewer side effects, for example. And the third research question is that you want to compare 2 or more treatments, so different language interventions in aphasia or, usual care, and then usual care with an add-on therapy. And the fourth research question would be, to examine dose response relationships. And the different research questions are basically associated with different control conditions. And for the first research question, where you want to find out whether the intervention works compared to no treatment or placebo treatment, you have an inactive control condition, basically, but that's, ethically problematic because, in many parts of the world, no treatment is not an option anymore for randomized controlled trials because you're not allowed to withhold potentially effective treatment from participants. So they have to have access to treatment at all times, during trial enrollment. And so more feasible is that you compare your experimental intervention to a waiting list control condition, which has the advantage that the control group also receives, the new study intervention. So it's also less frustrating for the participants. I'll talk a little bit more about this later. And then your second research question is that you want to compare whether your new intervention works better than usual care. And usually, the usual care should be the gold standard of care, but I don't think we have that yet in aphasia rehabilitation. And so in aphasia trials, we usually define usual care as whatever care is available in routine practice for our participants. So it's not really a gold standard. And the third research question is that you want to find out whether your new intervention is as good or better than an alternative intervention. For example, you compare your new intervention to an active comparator, which includes an attention control or an add-on therapy. And frequently in such design, you add a third arm, which is usual care control, because you also want to see whether your intervention is generally effective. As I said, fourth research question is whether a higher dose works better than a lower dose. These, are very interesting research questions for aphasia research. But if you want to do such a study, you run into the issue of having to recruit very high numbers of participants. And so no one has yet volunteered to do such a study. There's an ongoing trial where first estimates will be collected in the USA, but haven't heard of the results yet. So to summarize, you have a choice of control condition, which is, dependent on your research question, whether you want to find out whether your treatment works in general, whether you want to compare different treatments, or whether you want to study dose-response relationships. And when talking about control conditions where the participants do not receive a study intervention, you face a risk of finding negative outcomes that are experienced by the participants not receiving treatment because they may be frustrated about not receiving the study intervention. So it's basically the negative counterpart of placebo effect. It's called a nocebo effect, where you observe decreased performance in the control group. And this decreased performance basically explains your group difference. So the frustration may also additionally result in selective loss to follow-up. So you may lose more participants in your control arm, which, is not what you intend to. And I have an example here from a recently published trial, which, I think very nicely shows that obviously, there's a nocebo effect present in the data here. The groups did not statistically differ, but still you see, a quite obvious decrease from pre-intervention to post-intervention in the usual care group. So something is in here to show this. And I compiled this slide to show you, what the different control conditions are controlling for. So no treatment, for example, controls for spontaneous recovery, repeated assessments, and social attention by the research staff, but not, for the other factors that I mentioned. And the best control is when you have an active language comparator. So, for example, alternative aphasia intervention, but the problem is that you basically control for so much of the variance that you may underestimate your treatment effect, because you basically, control, for the language ingredient in your experimental intervention to a certain degree. So then may not be a lot of variance left. So my recommendation for the time being would be to use a combination of a waiting list control with usual care. And, it's a good question if this is maybe the most efficient control condition in aphasia trials. At least you can be pretty sure that you're controlled for most of, the unspecific treatment effects that you can expect. At the cost of overestimating your treatment effects a little bit, but, in the aphasia population where we have, where we expect small to medium effects only, I think it's ethically more appropriate to overestimate, to a reasonable amount than underestimate the treatment effect, and then, basically limit access to intervention, to treatment for people with aphasia. So there's a very useful tool which has been published by the Stroke Recovery and Rehabilitation Roundtable, which helps you to find an appropriate control condition for your randomized controlled trial. It's called the Consign Decision Support Tool. I've used it, it's very good, but of course, you still have to, make your own choices. So it doesn't, make the decision for you which control condition to use for a trial. So the last thing I want to discuss today is outcome assessment. So, how do you find out if the treatment was successful? And we published a tutorial paper last year, members of the CATS Trials for Aphasia panel reached international consensus on the best way of operationalizing treatment success in aphasia rehabilitation. So you can look at the paper for more detail. I just want to briefly summarize what the requirements for outcome measurement instruments are. in aphasia rehabilitation. They should allow standardized assessment, and preferably the assessor should not be involved in the therapy of the participant. That also is something which I think should be should be realized in clinical practice, that you shouldn't have assessor who did not treat the patient just because, you will have a more objective assessment. Your outcome measurement instruments should be have been evaluated in the relevant aphasia subpopulation. But they should be psychometrically sound according to accepted guidelines, for example, the Cosman criteria of patient-reported outcomes. And your outcome measurement instrument should be suitable to assess treatment priorities, which are relevant from the perspective of people with aphasia. And Sarah Wallace, organized the consensus on an international core outcome set, the Roma core outcome set, which is basically a unified minimal set of standardized outcome measurements instruments for the aphasia population. And these measures assess treatment outcomes which are prioritized by people with aphasia, and have listed them here. People with aphasia want, treatment outcome to be assessed in the domains of communication, general language, quality of life, emotional well-being, social participation, and also, self-reported treatment success and treatment satisfaction. And the measures that you can use, to assess these top treatment outcomes are listed here. Of course, there may be variations in other languages if you don't have the measure available, but they should be standardized in the aphasia population, as I said. And we have consensus for the measures on general language, quality of life, emotional well-being, and on communication, but we have not yet consensus on how we should measure patient-reported satisfaction with treatment or patient-reported impact of treatment. But we made a suggestion, we made a recommendation in our operationalizing treatment success tutorial paper. And Sally Singelman from Brisbane in Australia recently published the first qualitative data using, this grading scale that we suggested, that you basically should simply ask, your participants, how much has your, for example, language or communication or whatever your outcome is changed since you last did this test. And then you have a six-level Likert scale, Likert scale with the labels much worse, slightly worse, no change, slightly improved, much improved, and completely recovered. And Sally basically asked which scale level reflects treatment success from the perspective of people with aphasia and therapists. This was a very small trial, but you can see that, slightly improved is basically an indicator of patient-reported treatment success, particularly if you assess communication. There's high consensus that slightly improved is sufficient to count as treatment success. I also want to urge you to use aphasia-friendly versions of outcome measures whenever they're available, or you may also want to develop some new ones. Examples are shown here for social participation or quality of life. I included the QR code so you can find them. And I briefly want to touch individual treatment success or treatment responder rates as well. I'm a great advocate of group level results because they show you whether the intervention works for the whole population of aphasia, of people with aphasia. But it's also important to report, how many or the fraction of participants that show the treatment success on the individual level. And you do this by calculating benchmarks for individual treatment success. I won't go into the details, but you can read about the procedures in our paper. So we don't have information on minimally clinically relevant change scores at the moment in aphasia rehabilitation, but for many outcome measures, we can calculate the minimal statistically significant core change, and then that helps us to calculate which percentage of participants responded to the treatment. And Maya Minahimi-Falco published, benchmarks for various outcome measurement instruments in aphasia in her systematic review of maintenance following intensive therapy programs in chronic aphasia. And I list some of the benchmarks here for you. And there's an error in the original publication for the English SAQL, and the correct benchmark is listed here, and there will be a corrigendum published soon. So last thing I want to touch is what is a sufficiently powered trial, because you're required to include a power statement for your RCT in your publication. And therefore, it's very important to list the primary outcome of your trial. And it's preferable, I think it's preferable to have only one primary outcome measure, because otherwise you have to statistically control for multiplicity of testing. And you should state which, group difference you expect, also include the standard deviation that you expect for the group difference. You should list the type 1 error, so probably your significance level, and this also should be two-sided in all trials. You should list the assumed statistical power that you use to detect group differences, or that you assume to detect group differences. And then you should list the sample size that you calculated is necessary to achieve this desired precision. And from my own experience, I can share with you that the sample size will be at least 70 participants per group, just because, responses are so highly variable in the aphasia population. And all trial plannings I have been involved in, it was always, 70 or even larger sample sizes, sometimes even in the three digits per trial. And I list, some resources here for you where you can calculate, effect sizes, or you can estimate sample sizes for your trials. So one advice I have for you is to empower your trial based on treatment effects in a different aphasia population, because, we were basically forced to do this in our own trial, which I want to share with you today, because we have based data on a study which was conducted in people in the earlier stages after stroke, so in the late subacute stage, so I think 3 to 4 months after stroke. In our study was in chronic aphasia patients. So of course, we didn't achieve the same numerical group difference in the chronic stage, but we were lucky enough, that our standard deviation was significantly smaller, and so our trial was fortunately still sufficiently powered to detect the group difference. But it's very important, to find a priori information that fits your own population. So don't forget to register your trial, because reviewers will ask for it, and all medical journals basically require you to pre-register your study. And if you change anything while the trial is ongoing, please update the trial registry, because the journal reviewers are required to check the trial history when reviewing the paper, and they will notice any deviations from the original study design. And the Trials for Aphasia panel has compiled resources for planning randomized controlled trials. And so many of the information I shared with you today can also be found here. So this is very useful. And we also, I have a section on patient and public involvement, a topic that I couldn't, discuss today because of the time limitation. So I want to briefly summarize. If you want to please, reviewers of medical journals, do conduct a randomized controlled trial to find out whether your treatment works, using the key principles of RCTs. Select a control condition or an RCT design that is appropriate for your research question. State your PICO question explicitly. Choose a control condition that is adequate for your research question, but that also maximizes your probability to find at least moderate treatment effects. Use standardized outcome measures, preferably the ones also included in the ROMA core outcome set, but also assess meaningful outcomes from the perspective of the patient, of the person with aphasia. If you include additional outcome measures, they should be standardized in the aphasia population and have sound psychometric properties, and do report, blinding, your blinding procedure and the guessing rates. If possible, use blinded endpoint assessment for your primary outcome. To adequately power your trial for the primary outcome, and please register before recruitment starts. And so that was it. I'm sorry for being a little bit over the time, but thank you very much for your attention.
Thank you, Caterina. Trials are, as I've learned, extremely complicated. So thank you for taking us through some of those key aspects. If people want to put any questions in the chat, or alternatively, if you raise your hand, I'm quite happy to unmute people to ask their question. But I have plenty of questions for you anyway. I guess one is that in order to do a trial really well and follow all of those recommendations, the number of resources or the amount of resource required is quite high. Do you think some of those are more important than others, or like would there be a ranking system for those features, I suppose?
Yeah, this is a very good point. I forgot to mention it because these trials are very expensive, and particularly with a waiting list control condition, you basically double the number of participants that you have to treat in your trial. And if you're paying on a case by case, if you have case by case payment in your trial, that of course doubles your cost. And for the trials that we are submitting in Germany now, randomized controlled trials, 2 arms only, 70 participants per group, we're coming close to 3 million euro at the moment. And of course, it's very difficult, to get funding, for such costs, because aphasia is, research is a niche research area in medicine, and so it's not a top priority. But my advice would be, don't save money on biostatistician, because it's very important that you have someone who assists you with trial methodology, and everything that has to do with the therapy, not frustrating patients. So I think you should also spend the money on a waiting list control condition, to minimize the risk of finding nocebo effects. As I said, we already expect only small to medium treatment effects in our research, and you don't want to lower this by frustrating participants.
I hope my audio is okay, but there's a tremendous amount of rain coming down right now. Nelson Hernandez is asking, he said, thank you for sharing such important information. My question is related to Likert scales. Is a consensus on what level Likert should be used, or which is more sensitive to change? Is my audio dropping out?
Likert scales. Okay, Likert. Okay, then.
So how many levels might that need to be?
Yeah. Yeah, well, no, this is a very good question because, when we were developing this paper on operationalizing treatment success, looked into this, and there are publications on, the optimal levels a Likert scale should have, to minimize the risk of response bias. And I think it was, 5 to 7. So definitely 6 was, within this optimal range. And the scale is not balanced. And, we already had viewer comments because, we have, a minus 3 level where you say, that you are much worse. And, the positive pole on the other side, the positive extreme would have been that someone, no, it's the other way around. So we have a very positive scale, +3, so completely recovered, because it can actually happen, early after stroke. But, we don't expect that anyone will completely lose, language abilities through therapy. And so we thought it was a little, ridiculous to include it in the scale.
Well, if they get that much worse, perhaps they need to go and see a neurologist anyway. Another question I had is about usual care. So if we look at the VERSE trial in Australia, the usual care arm had a surprisingly high dose. And my theory is that because the clinicians were being observed, they just, maybe not even consciously, but gave them a little more than they normally would. So do you think usual care should be controlled or prescribed, or should it be completely natural? Does that make sense?
Yeah, it makes sense. I mean, of course, it's much better if you're able to control, what kind of therapy dose people in the control group get. But I mean, usual care, as I said, in theory, should be a gold standard. And, it would be nice if everybody in routine care gets, the same dose, because, the research suggests that they should get a certain dose, for example, or a certain type of a therapy. But I think it will be hard to control, because I said you can't withhold, therapy from patients ethically. So basically, they're allowed to receive any kind of treatment they can access. So I'm not sure whether you're able to control it. But I agree, as soon as therapists know that their participants are in the usual care group, they may be extremely motivated, to do a particularly good job, and, even, do more therapy than they would usually do with their participants.
All right, I think I'm going to squeeze in one more question, if that's okay, just before we hit the hour. You recommended sort of block or stratified randomization. Is the recommendation not to have simple randomization where you just randomize each participant?
Well, I mean, the blockwise randomization is particularly done because, you have to do a multi-center trial. You can just this is not a requirement for a randomized controlled trial, shouldn't be a single center. And then you basically, within each of the centers, you want the same number of participants in each group, allocated to each group. So that's another reason why I do blockwise. But even within the centers, for example, if your first block is 4 participants, and you have, once this block is filled, you have 2 in each group, and then the computer would randomly decide what the next block size is, so that investigators can't guess, have no chance of guessing, what the next assignment will be. Because, you may have participants who say, I will only, take part if I'm in the experimental group. And so if the experimenters have any guess what the next assignment will be, that will be huge bias.
Did you say it must be a multi-center trial? Or is that only because of the
well,
numbers
if you want to have a level 2 study, it's a must, because you have to show that it's not only working in one context, basically. Yeah, so
okay.
multi-center is another requirement of a sound randomized control trial.
Right. Well, thank you for your time today in presenting.
Yeah,
I'll just
thank
let
you
everyone
very
know what's
much
happening
for.
next. So if you do want to revisit today's seminar or share it with others or look at some of those QR codes that came up, the recording will be available on our YouTube channel in a matter of a few weeks, along with many of the past seminars. And to stay informed about our work, please follow our blog or Facebook or LinkedIn. And that way you'll receive updates on our research, recruitment opportunities, and many other resources. So please follow us there. For 2025, I'm pleased to say that the seminars will continue. So we're currently lining up some fascinating topics from aphasia researchers in Australia and internationally. So follow us along there to hear more. Thanks for joining us, and thanks again, Dr. Breitenstein.